Assalamualaikum and good day to our viewers. In this video, we are going to present to you about an article titled Essential Oils from Fennel Plants as Valuable Chemical Products, Gas Chromatography, Mass Spectrometry, FTIR, Quantum Mechanical Investigation, and Antifungal Activity. We are from RAS 1205C and our group consists of 4 members, which is me, Nur Ali QISTINA binti MOHAMAD NAJIB. The next presenter will be Nur Ali SOFIA binti ZULKIPLI. The next one will be Nur AFIQAH binti MOHAMAD HANIZAM. And lastly, Nur FARAH QISTINA binti MUSTAPA. Essential oils are complex natural mixture containing many bioactive chemical compounds. To fully understand their chemical composition, reliable analytical techniques are required. The analytical method used in this research is gas chromatography, mass spectrometry, Fourier transform infrared spectroscopy, and DFT and MNDO. However, in this presentation, we will focus only on gas chromatography or GC component. The main purpose in this study is to separate and identify the chemical compounds present in fennel essential oils. The aim of gas chromatography in this research is to analyze the chemical composition of fennel essential oils, separate the individual volatile compounds found in the oils, determine major bioactive components, and compare the chemical composition of oils obtained from 2 different parts of the fennel plant, which are the fennel leaves and the fennel umbels, which are the flower clusters. Through GC analysis, the researchers were able to identify which compounds are the most abundant in each sample. For the GC analysis, the sample used were essential oils extracted from fresh fennel leaves and fresh fennel umbels. Before gas chromatography could be performed, the oils were first obtained through an extraction process. The extraction method used was hydrodistillation with Avenger apparatus. After extraction, the essential oils were carefully stored and prepared so that they could be analyzed using gas chromatography. This introduction provides the background and purpose of using gas chromatography in this study.
Now, I will explain the gas chromatography methodology used in this study. First, under GC analytical method, gas chromatography mass spectrometry or GCMS was used as the analytical technique. The analysis was carried out using a thermoscientific GCMS system. In this method, the GC component play an important role in separating the volatile compounds present in the essential oil sample. Next, I will explain the function of the GC component. The GC was used to separate volatile compounds inside the GC column. During this process, separation occurred based on rotation time and a chromatogram was produced for analysis. Each compound emitted at a different rotation time depending on its chemical properties. Then, we move to the operating conditions. A carrier gas was used to transport the sample through the GC column. Temperature programming was also applied to control the separation process. This condition helped to achieve effective separation of compounds with different volatilities. Lastly, for the identification of compounds, each compound was identified based on its radiation time, which indicates when the compound emitted from the column. The peak error percentage represents the relative amount of each compound present in the sample. Each peak observed in the chromatogram corresponds to an individual volatile compound. Next, I will explain what gas chromatography measures. First, under the role of gas chromatography, GC is used to separate a complex mixture into individual components. In this study, it allows us to separate and analyze each compound present in the essential oil in detail. Next, we move to the data produced by GC. The instrument generates a total ion chromatogram or TIC. This chromatogram displays distinct peaks for different compounds. Each peak appears at a specific rotation time, which is unique for each compound. Lastly, under information determined by GC, gas chromatography determines the relative evidence of each compound in percentage form. This is done using the peak error percentage to compare compound quantities. From this data, the chemical profile of the essential oil can be established, providing an overall overview of the volatile composition of the sample.
Next, let's look for the results and discussion. The GCMS analysis identified several bioactive chemical compounds within the essential oil of fennel plants. The study compared the chemical profile of oil extracted from the leaf versus those from the umbels, revealing significant differences in their primary constituent. The study found that while the same major compounds were present in both parts, their concentration varied significantly, first in anethole, second estragole, third D-limonene, and fourth French horn. From these results, it can be concluded that leaf oil is rich in anethole and estragole, which are important aromatic and bioactive compounds. For fennel umbels essential oils, the GC results showed a different distribution of compounds. It can be concluded that umbels oil is dominated by estragole, with higher French horn content compared to leaf oil. Moving on to the importance of our GC results, the gas chromatography analysis was highly successful because it allowed us to take a very complex mixture, the fennel essential oil, and separate it into individual poor components. This is crucial because it allowed us to identify the specific bioactive markers responsible for the plant's properties. Most importantly, the GC data prove that the leaf and umbel oils are not the same. We see that fennel oil is primarily composed of aromatic compounds and terpenes. We notice that the concentration of these molecules change depending on which part of the plant we analyze. For instance, the high levels of anethole and estragole found by the GC are what give fennel its unique smell and, more importantly, its strong medicinal and antifungal activities. This tells us that the quality of the oil depends heavily on the ratio of these specific compounds.
Next, we move to advantage of gas chromatography. Why gas chromatography is important in this study? It is because it allows accurate separation of the complex mixture in phenols and essential oils. It provides fast and reliable analysis of the chemical components. Gas chromatography also enables the identification of volatile organic compounds present in the essential oils. In addition, it provides a detailed chemical fingerprint that helps compare the chemical composition of different plant parts. Next, we move to limitation of gas chromatography. Despite its advantage, gas chromatography has some limitations. It is only suitable for volatile compounds and cannot analyze non-volatile substances. Gas chromatography analysis also requires proper sample preparation to obtain accurate results. Next, we move to antifungal activity. Gas chromatography analysis revealed that fennel essential oil contains several bioactive compounds responsible for antifungal activity. The major compounds identified by GC include transanisole, estragole, fenchoans, and D-limonene. Among these, transanisole was reported as the dominant compound in many fennel essential oils chemotypes. Estragole-rich essential oils show stronger antifungal effectiveness, especially when combined with other components. Fenchoans contribute moderate antifungal activity and enhance fungal growth inhibition when present in higher concentrations. Limonene also play an important role by supporting the antifungal effect through synergistic interaction with other compounds. Lastly, for the conclusion, gas chromatography was essential for analyzing the chemical composition of fennel essential oils. It successfully separated and quantified the major compounds present in the oils. Gas chromatography analysis identified anethole and estragole as the dominant components. It also demonstrates clear chemical difference between the leaf and umbels. These results provide a strong foundation for further biological and antifungal studies. Overall, gas chromatography was the main technique used to analyze chemical composition, compare different plant parts, and determine the bioactive constituent responsible for antifungal activity.
This is our reference.